Good morning, children. This is my third video on our lesson, the circulatory system. In my previous video, I had completed the comparative table between three cellular elements, RBCs, WBCs, and the platelets. After that, I discussed about different types of WBCs under granular category and the non-granular category, and we ended our class with important terms like diapedesis, phagocytosis, and pus. Today's new topic to begin is blood clotting or coagulation. See, normally in a body, when there is rupture of a blood vessel, the blood starts oozing out. So, in a healthy body, the process of clotting, various reactions that we are about to study, should begin internally between 2 to 8 minutes. But that does not mean that 8 minutes the clot will be seen on the wound site. No, it will be seen only after 3 to 4 days, right? But the process internally begins between this 2 to 8 minutes. But there are patients of certain ailments like hemophilia, liver diseases, and malabsorption. Hemophilia is a genetic disorder in which there's a clotting factor 8 that is lacking, right? So in these patients, the clotting time is abnormal and much delayed. So that leads to profuse loss of blood. Let us start with the steps in blood clotting under 4 very simple steps. The first step is the injured tissue cells and the platelets, they disintegrate at the site of wound and release a substance known as thrombokinase, also called as thromboplastin, and more recently we have given the name Stewart factor or factor X. Alright, so normally this is not present in our bodies, right? But this is formed only after the broken down platelets and the injured cells. Alright, now step 2, the same thrombokinase which has formed, right? It acts as an enzyme, and with the help of calcium ions of our plasma, there is a substance which is inactive. We call it as prothrombin. Pro means inactive. So this will be converted into an active thrombin. So remember, children, one thing what is very special about these reactions is whatever is formed here on the right side will be used up on the arrows in the preceding steps. So here what is formed will be the next enzymatic substance here in calcium, presence of calcium ions, converting prothrombin, an inactive substance, into active thrombin. Step 3, now here what is formed again comes on the arrow. So step 3, this thrombin in the presence of calcium ions, right, it will react with a soluble substance of the plasma. We call it as fibrinogen, and the same would be converted into an insoluble sticky thread called fibrin, right? And these threads, they form a mesh or a network at the wound site. All right. Then the fourth and the last step is when the blood cells are trapped. See, these threads that are formed, fibrin, they're sticky. So whichever blood cells they come in contact, they will be trapped. Now the threads, they start shrinking and they squeeze out from the rest of the plasma in the form of a clear liquid called serum, right? And when the liquid has been cleared out, the solid mass that is left behind will be called as a clot or thrombus. See, I'll show you by a diagram given at textbook page number 92. In this diagram, what is being shown? 2 test tubes are shown, the left and the right one. In the left test tube, the blood is taken, right? And soon what will be observed is that a clot is formed in the usual way and the serum is squeezed out. All right. So the serum that is squeezed out from the clot will collect on the surface of the contents in the test tube, right? You can see, in other words, what we can say is here serum, the fibrin threads and corpuscles are all mixed up. But the same after some time. Remember, children, of the total blood sample, if we remove these threads and the clot and the RBCs, WBCs and platelets, the leftover substance is called as serum. That means serum does not contain any threads, or it also does not contain any blood cells. And this is a clear substance, right? So blood is a mixture of the same serum, the fibrin threads and the corpuscles. Corpuscles means all three types of cells. All right. Same thing is indirectly explained over here, that here when we are saying the fibers, they are shrinking and they are squeezing out from the rest of the plasma along with the blood cells. Whatever is left will be now called as serum, and this solid mass will be left behind as a clot or a thrombus, right? Now some questions to understand this 4-step mechanism of blood clotting can be definitely asked in your council examination, but there will be some relevant questions too. Like for one example, what happens if the platelets number, they fall down to an abnormally very low count? In this case, coagulation may occur very slowly, but this often leads to a condition called hemorrhage, which may lead to death of the person. Same thing is commonly the reason of death in patients of dengue fever. Dengue fever, which is caused by a virus which has taken hundreds of lives till today, right? And not just hundreds, we can say thousands of lives of our country. And this started since 1996 till date, that we can say. And there is a same fatality, what we can say, due to hemorrhage leading to death. Another question can be said that, is it true that some people believe that exposure of the wound to air helps in a faster clotting process, or that the clotting depends on the exposure of wound to the air? This is totally wrong. It's a myth. In fact, clotting can be caused by the movement of blood over a rough surface, like cholesterol deposit on the inside of a blood vessel. So, to sum up, this is absolutely wrong, and this has nothing to do for the clotting process to come in contact with the air. Alright, so this is all for just now, children. The topic is small. My next topic would be on blood groups, right? Here, I am sending you only one single page for you to write down, and you may definitely read page number 92, the entire process of clotting, along with the flowchart that is explained. Same thing what I have given you in the reactions. It has been explained in the form of a flowchart here, along with the definition of serum. That's all for just now, children. In my next video, let's start with the process of blood groups later. Thank you for just now.